to now announce the next presentation, which will deal with genome modification, focusing on the CRISPR-Cas system and the great potential that this technology has for potential application in plant breeding. The presentation will be given by Holger Puchta, who is a professor at the head of the Puchta Institute at the Karlsruhe Institute of Technology. Mr. Puchta actually comes from biochemistry, and so he has worked at the respective Max Planck Institute in Munich and at the Friedrich Institute in Basel before moving to IPK. And in 2002 he then accepted the call to Karlsruhe, where his research focus now lies on DNA modification and genome engineering. And his research, I may say, is highly successful. There are two big European Commission grants that were given to Mr. Puchta, and also numerous publications are proof of that. So we were obviously very happy when Mr. Puchta accepted our invitation to talk about his work here today. Please welcome.
So, I want to thank especially the PhD students for inviting me. And I think it's always a special honor to be invited by PhD students. And therefore, I will also give my talk on this side of the room, because here are the PhD students sitting that invited me, and not in front of the big professors. I hope you don't mind. Okay, so I gave a very general title, Genome Engineering in Plants, Past, Present and Future, and I have to tell you that I will not go back 200 years and I will not speculate about the next 200 years. Genome engineering, I will cover the last 25 years, because there is indeed a history of 25 years of genome engineering, and I will speculate what we might do in the next 10 to 20 years. Okay. So, what I'm telling you about is in the first part a little bit about the past, and then I will tell you about CRISPR-Cas systems, mainly systems we developed in the lab. I will tell you about mutation induction. I will tell you about gene targeting, inducing predefined changes by homologous recombination. And then I think this is a very good audience also to touch the topic of regulation. I will give you a little bit of overview of the current state of regulation. I hope we all don't get too depressed about that. And then, nevertheless, there is a future, and my take-home message is already now that there is future, and I will tell you, I will speculate a little bit what we can achieve in future with this fantastic technology. Okay. I thought, as I'm not a breeder but a biochemist, I at least should in the beginning talk a little bit about breeding. And historical breeding brought us many monsters. What you see here are very useful monsters that all came out of brassica. And the point is, of course, in this process of selection, you lost a lot of genetic variability from brassica. So now we end up with cauliflower or with broccoli. But the problem is, of course, for breeding, and nowadays you cannot go back and you cannot, sorry, I have to learn this first, you cannot really transform a broccoli by breeding now in a cauliflower or a cabbage. So in principle, one of the major problems of breeding is a loss of genetic variation. And this loss of genetic variation was a problem for a long time in breeding. And breeders came up with the solution. And one of the solutions they had was actually an extreme brute force solution. And the solution was, if we want to change genetic material, why don't we just put harm to genetic material? And the most efficient way to harm genetic material is to induce double-strand breaks. And if you induce a double-strand break, you get a repair process. The genetic information is restructured to a certain extent at the break site, so new mutations arise, and this gives you variation. Most of the time, this process is not leading to something valuable, to a valuable trait, but sometimes, in some cases, it is. And that's why this technology was really adopted by breeders a very long time ago already, in the middle of the last century, of the last millennium. and it was used just by using X-rays to induce mutations. There was also the approach to use chemicals, genotoxins, to induce mutations. But in principle, this is a very common way to really produce new genetic variation in new traits. And this technology is it's being applied in plant breeding for the last 70 years. And the problem is, of course, because you induce a lot of random mutations, you get a lot of this so-called off-site effect. So, in principle, you have once in a while a good mutation, but most of the time, most of the mutations are adverse, and so you have to cross out all these adverse mutations till you get the good mutation. This kind of technology requires an ample of time and an ample of screening, and I don't have to tell you the details because you are the breeders, I'm not the breeder. So this technology nevertheless worked very well, and there are many examples now worldwide, many cultivars that have been produced by this technology. All European short-stem barley was produced by this technology. There are more than 3,000 different cultivars worldwide grown that are produced by this technology. A very prominent example, for example, is also this grapefruit here, Ruby Red. This here, the seeds, what you can see. Also here we have the buckwheat as an example. Okay, so this technology is extremely successful, but it's also extremely laborious. And therefore, since a long time, and now I'm starting with the story of genome engineering, since a long time people were thinking about, can we do this better? Are there other ways to induce breaks? And of course, the change of covalent linkage of genetic information is a general problem in nature all the time. And how does nature achieve this? I mean, if you look, for example, at inheritance, meiotic recombination, here you see the chromosome of the mother and the father interact with one another, and this exchange of genetic information during meiosis, the first decisive step is the induction of a double-strand break. Only if double-strand breaks are induced, this exchange of genetic information can occur. And there are many other processes in nature where the first step is always, if you want to change genetic linkages, you have to induce breaks. And for this induction, enzymes are used. So in principle, it was clear if we can get hold of enzymes that induce double-strand breaks at specific sites in the genome, we will be able to manipulate genomes. So the big question was for a long time, are there enzymes that are specific enough to really induce a single break in the genome? If you think about all the restriction enzymes you always use in the lab, like ECHO1, which is a 6-mer site, so if you throw this on a plant genome, it will chop up the plant genome completely. What we need are enzymes that only cut once statistically in a plant genome. And indeed, the story began a long time ago, 25 years ago, when people became hold of an enzyme from yeast that was specific enough, it was called IC1, that you are able to induce double-strand breaks. So in principle, that's what I told you before, we need to have enzymes that are specific enough that they only cut once in the human plant genome. And here is the respective publication. And the guy who first discovered this enzyme was Bernard Dijon. And we profited a lot from him because he gave us this enzyme. This is an enzyme, this is a homing endonuclease, which he isolated from mitochondria of plants, and this gave us the opportunity. It had an 80-mer restriction site to, for the first time, analyze what happens if you induce a specific break in the plant genome. At this time, I was a postdoc at the Friedrich Michel Institute, and in principle, having this enzyme in my hand and being the only person worldwide working with this enzyme for the next 10 years was the guarantee of my own scientific career. The problem at this time was that really nobody was interested. So I felt a little bit alone on a hill, but in the last 5 years it's changed dramatically. Now I feel like in the middle of a tsunami. Okay, so what could we achieve with this unique tool? We could really ask now the question, what can you achieve if you induce double-strand breaks in the plant genome? All the different things that thousands of people do now, also with CRISPR-Cas. So, one major problem was at this time that people tried to integrate at specific sites via homologous recombination DNA molecules. And for this in plants, because homologous recombination is working extremely inefficient, they had to make hundreds, thousands of transformation events, which was absolutely impractical. And so we asked ourselves, because all the recombination models said you need a double-strand break to induce homologous recombination, what happens if we induce a double-strand break and offer the plant genome a tDNA with homologies here and here that are also there in the genome? And we made this experiment and we realized we can induce homologous recombination by orders of magnitude if we use such breaks. So we came in the percent range. This was good news at this time. Bad news till today is we are still fighting, even nowadays, with this percent range. That will come later in my talk, tell you a little bit about current solutions we are trying to achieve. So the question is, of course, if homologous recombination is so inefficient that even if you induce a break, most of the breaks are not repaired by homologous recombination, what is the main mechanism? Well, the main mechanism is non-homologous end joining. So we could show indeed that there are, maybe this is not so relevant for you as breeders, but as I'm a molecular biologist, I'm always happy with mechanisms, that there are even two different mechanisms. One is called the classical non-homologous end joining. So the two ends are put together, sometimes one or two nucleotides are lost. If you have an open reading frame, if you use one or two nucleotides, the open reading frame is destroyed. And there is this mechanism, alternative end joining, where short microhomologies hybridize with one another at the ends. So the other parts are chopped off, these are bigger deletions, and most of the time, of course, you destroy an open reading frame if you have a break in the open reading frame, so this is highly mutagenic. And this is the reason why, if you induce a break with CRISPR-Cas nowadays, in plant genomes, most of the time you can induce mutation in open reading frames. Okay, we also found a few extremely surprising things. And I want to mention this because these are things that happen if you induce double-strand breaks. This is biology, and it has nothing to do with the CRISPR-Cas system, because sometimes people say, oh, the CRISPR-Cas system is not working very well. No. Everything that you can induce with the CRISPR-Cas system is classical double-strand break repair. And there are other things that can happen. So what we also found out, which was at this time extremely stunning, was that tDNAs can integrate into double-strand breaks even without homology. So sometimes you will find this out in experiments that you now perform with CRISPR-Cas, that DNA from outside is integrated into this break. And by now we understand why, because we think this is the mechanism how tDNA integrates. I mean, all the DNA apparently is recognized by the cell as damage, and if there are open ends, it's just linked to DNA damage. For us at this time, this was very surprising. Now it's generally accepted. And another interesting thing you also find is that sometimes you find not sequences from outside the genome integrated into the break, but even as are sequences copied from other side of the genome into the break. So this is also a common mechanism you might also find if you use CRISPR-Cas induce breaks now in genome engineering. And what we could find at this time in tomato, this is, by the way, a Southern blot. I don't know if many people in this room know what a Southern blot is, but in principle, the elder might remember, so you can see the number of sequences in the genome you have to hybridize with a probe, and this is in principle 5 different Southern blots, and what we really could show here is that we found integrated into the genome repetitive sequence, but also unique sequences. So in principle, whenever you induce a double-strand break in a genome, you have to be aware, besides the things you in any case will get, with high probability, there are certain pathways you also switch on at low probability that gives you other results. So you always have to control what you get if you use a double-strand break. Okay, we also asked the question, what happens if you induce 2 breaks at the same time? If the 2 breaks are in close proximity, this you can see here, then you can cut out sequences out of the genome. At this time when we published this, we made a patent together with BASF because they wanted to cut out antibiotic resistance genes out of transgenic plants. And later on, we also induced breaks in different chromosomes. This is Michael Pacher, one of my former postdocs. And in principle, and I think that was a very important experiment we could show, and I think this was also important still for the future of genome engineering, that you can indeed induce chromosomal exchanges, translocation, reciprocal translocation between chromosomes if you induce breaks at 2 different chromosomes. So this was all done with this cute enzyme IC1 a long time ago. The trouble with this homing endonucleases was, of course, that they had one specific site in the genome and you couldn't address other genes. So in principle, there was a big effort taken by many groups worldwide to transfer the technology to other kinds of enzymes, artificial enzymes that would also be able to target other sites in the genome. So in the beginning, people started to manipulate homing endonucleases. This was not very successful. Then they used transcription factor binding motifs of zinc fingers fused to a restriction enzyme, and they called it zinc finger nucleases. And in principle, we were able to really induce breaks at other sites in the genome using the zinc fingers with low efficiency. And later on in 2010, transcription activator nucleases were developed. So this is a DNA binding motif. This comes from a pathogenic bacterium of plants that is used to recognize specific sites in the genome, and this specific domains can be mutated in a way that other nucleotides can be recognized. So all this was happening quite some time ago. So you see in principle, the technology, what we could do before, is not as new as you might think. But in principle, the big change, the availability of the technology in every lab only came with the CRISPR-Cas system. And this brings me to CRISPR-Cas. And what the hell is CRISPR-Cas? I just want to remind you, most of you might know. So this is indeed a bacterial system. People knew that for a very long time that there are so-called CRISPR array repeats in the genome. These are arrays which you have the same sequence disturbed by sequences of other things. And you have a protein coding region, the Cas locus. And only after a long time of research, people realized that these repeats are transcribed into RNA and then they are processed. And indeed, the small specific pieces are pieces from pathogens, from virus DNA or from plasmid DNA. And these guides are then going together with the protein. And what you can then obtain is a nuclease that recognizes specifically, it's defined by this 20-mer sequence within the RNA, specifically other DNA molecules, because this was stored from the infecting virus. If a virus, or better said phage, comes in again the same phage, then this can be recognized and in principle can be destroyed. So it's a bacterial immune system, and it's very, very generally distributed in bacteria system. And it took us, it's in principle, if you think about scientific developments, because we are also thinking about scientific developments in this room, it's astonishing how long it takes us to understand the bacterial system. So the bacterial systems were mainly researched in the 70s and 80s of the last century. And for me, it's hardly understandable that it took us so long to find out how the system works. And the most decisive thing was really the work of Chapuquier and Dudner in 2012, where they could show, yes, you can really make out of this an enzyme that you can very easily reprogram. You have this, they made a single guide RNA of this complex system, and the Cas9 protein, if this comes together, you have the most efficient kind of nucleases. Just by defining this 20-mer match here of the RNA, you can target it to all specific places in the genome. And I think this finding, there is no other finding, at least in this millennium, at least in the last 30 years, that is more revolutionizing biology than this specific finding. And I have to tell you a short story. I remember the day, it was June 2012, when the paper came out in Science. And I had 2 co-workers that came to my office and told me, Holger, there is a new enzyme. I mean, we were working for double-strand break repair since 20 years, so everything related to double-strand breaks was interesting to us. So they came to me and said, Holger, there's a new enzyme, we should try it. And I told them, I think this is total crap. This will never work. And it will never work. It will never work because it's a bacterial enzyme. Bacterial genomes are much too small, it will not be specific enough, and bacteria don't have a chromatin structure. And therefore, most probably, it will not recognize plant chromatin DNA. And so we decided not to work with the system. For half a year, I felt fine. The day before Christmas, I again looked into Science. And there were two publications showing by Feng Zhan and by George Church showing that CRISPR-Cas is working in mammalian cells. And then I thought, shit. And we lost the race. I mean, we lost the race. I thought we lost the race before we started. So in principle, fortunately, there was one big problem my plant colleagues couldn't solve. And this was the problem, and I have to admit, I'm working basic biology, so I'm doing weird things. For you, I mean, I'm working with a weed. So our model plant is Arabidopsis, the model plant of the plant community is Arabidopsis, so you don't have to get a heart attack now. For us, it's really the most wealthy plant we have. Okay, so in principle, nobody was able to induce mutations in the Arabidopsis. This was our chance. And we set up the system, and if you have a good PhD student, I had an excellent PhD student, Frieda Fauser, who then worked day and night and set up the system in Arabidopsis. So in Arabidopsis transformation, you make Excel transformation, you dip the plants with the flowers, I don't know whether you know this, and so you get transgenic plants, and we use this technology, and we used in contrast to everybody else in the field, not this 35S promoter, which is normally used for expression, but we used the ubiquity promoter. Everybody else used a 35S promoter, and nobody was able to get really mutations into the next generation. But with the UB promoter, we immediately got mutations in the next generation. So indeed, we could catch up. We were in principle the first to show that yes, you can use CRISPR-Cas to induce heritable mutations in the model plant Arabidopsis, and only heritable mutations are, of course, something that makes sense if you work with a model plant, obviously. So in principle, what I show here, these are the patterns you induce. We induce the mutations in different things. That's the ADH gene. Sometimes you have an insertion of one nucleotide, sometimes you have deletions. All these leads, of course, to the destruction of the open reading frame. This is another example, the TT40. Here we have an insertion of just one nucleotide. So this is the seed code of Arabidopsis wild type, and with this one nucleotide insertion, the seed code stays yellowish. So the system worked very nicely, and we then also checked mutation rates. Mutation rates in the first paper, which we published in the beginning of 2014, were about 15%. And we were very happy because it was extremely hard before to get mutants in Arabidopsis, and this was a big step forward. But we also thought maybe we could get a little bit better. But before I tell you the story, I just want to make a quick stop and compare this technology now to what I told you before, the classical mutagenesis. I mean, what we have now, and this is how most people use the technology just to induce mutation in this gene, we have a technology with the molecular scissors that is, we have much more experience, I think, as you might realize from my talk, than just the last 3 or 4 years. We have experienced it. With 25 years, we know very exactly what kind of change can we induce. The CRISPR-Cas system, in contrast to other classical mutagenic systems, is directed, and you hardly have any off-site effects. So the first enzymes used with CRISPR-Cas, they had a certain off-site effects. Now there are further programs going on to optimize these enzymes, and there is hardly any off-site anymore. And of course, the method is fast and efficient, you don't have to cross-back. So it's a tremendous step forward. And of course, this is already used now, especially in the States, already produce a lot of traits, and most of you know this. So some years ago already, powdery mildew-resistant wheat was produced, virus-resistant cucumber, drought-resistant corn, or tomato varieties with changed root development were shown to be reduced last year with this technology. I mean, this list is growing endlessly. There are many, many more traits. There are more traits coming every week, and sooner or later we will also see these traits brought out. Also, DuPont Pioneer has some traits which I realized I didn't put on the list that will be soon grown in the field. Okay, coming back to the point, we were at 50% mutation frequencies, and we thought ourselves, can we get even better? And I told you that CRISPR-Cas is a system where you find in many different bacteria. So in principle, there is not one CRISPR-Cas system, there are many related CRISPR-Cas systems, and so we also tested systems from other bacteria besides Streptococcus pyogenes, which is a general system, and if somebody in the room is using Cas9, most probably it's from Streptococcus pyogenes, and we tested other systems. This was work done by another PhD student, Janet Steiner. The setup was exactly the same as before. We all test this in Arabidopsis, and here are the results, and I only show you the results for aureus, because the work with aureus gave me what I would call a positive heart attack. You see here, we got tremendously high mutation rates of 90 to 100%. And it turns out that at least in Arabidopsis, the Cas9 of aureus is a much better enzyme. It's a real gold mine for us, and we by now only use the Cas9 by aureus. The PUM sequence, I didn't tell you details about the PUM sequence. That's the sequence you have that the protein is recognizing before it cuts, is a little bit longer, so there's a certain distraction to the system, but still the efficiencies are much higher, and the patterns are in principle very similar. You also get deletions that knock out genes, and in principle I also can recommend other people to try, besides the pyogenes Cas9, the aureus Cas9. We are also happy if anybody is interested to send you, even without MTA, the clone, if you want to have a try. I think that this is a real alternative, and recent biochemical study gave us also an idea. Why is this the case? Because apparently pyogenes Cas9 is sitting on the DNA after cutting and never going off, but the aureus enzyme is cutting and going off later on, so it's a multiple process enzyme, and that's why we think it's better than the pyogenes. Okay, talking about different things you can achieve using Cas9. What you can also do is, and this is important if you have different kinds of Cas9, you could also transform this nuclease into a binding protein by just knocking out the active amino acids and induce a break. And then, and I think this is very important, you might have seen this before, then you can target specific things to specific sites in the genome. For example, you can change epigenetic the chromatin structure. You can also make base editing by deamination of cytosines, or, and I want to shortly show this, you can also fluoresce mark respective sequences in the genome. And we did this, so we fused the Cas9 of aureus and the Cas9 of pyogenes to achieve P multimer and a ruby red multimer, and then tried this out together with Andreas Huhm, a good friend of mine, for visualizing respective sequences in the plant genome. In this case, we used guides that target to the telomeres, and you see without guide, everything is in the nucleolus, but with the guide, we have specific spots, and in principle, you can then use this technology for live DNA protein dynamics, so you can express this in live cells, and here we have the respective Cas9 fusion to telomeres, and here is a telomere binding protein that was also fused to GFP, and here you see the overlay. So in principle, this works very nicely, and the point is, of course, because we have now different enzymes from pyogenes and also from aureus, we can in principle mark different sequences at the same time in a cell, or more globally speaking, we can now on different levels also manipulate. So I think the point is, and I don't think that many people realize this already, that that you don't have only one kind of Cas9, you can have multiple kinds of Cas9, and you can progress them in multiple ways that you can really achieve very complex changes in the metabolism of plant cells. Okay, now I come to CRISPR-Cas mediated gene targeting. So what I told you before is mutation induction. You induce a mutation, you get different kinds of changes during double-strand break repair. But often you want to have one specific kind of change to exchange one specific kind of amino acid. And for this you need homologous recombination. So we developed a technology that was dealing with the problem of the low transformation efficiency. So I told you before we started this, you needed millions of transformants to have one targeting event. Then we showed, okay, if you induce a break and you make homologous recombination, you need still hundreds of transformation events. And so we thought, if you have a plant that is even transformable, this might be no problem, but most plants are not easily transformable. So can we find a situation where the job of transformation is avoided, or let's put it the other way around, where the plant is doing the job, not your technician or your PhD student? So, this is the answer. The answer is, yes, we can do this if we perform targeting during the life cycle of a plant cell. And therefore, we integrate the target vector before and only induce targeting and using double-strand breaks during the life cycle of the plant. So how does this should work? You induce breaks, and you induce it in the donor in the genome, and at the same time in the target in the genome. Homologous recombination takes place in the regions that have this homology, same color, and then the reaction takes place, and in the end you get rid of this donor by segregation or whatsoever. So in principle, this happens during the lifetime of a cell. You express your enzyme with a promoter that is developmentally regulated, and what you end up is in the end seeds, and you only have to screen seeds. So we set up this technology already, you see in 2012, this IC1, and then when CRISPR-Cas came around, we also adopted it to CRISPR-Cas. You can do in principle two things: either you can introduce a new gene in a specific locus, or you just can also exchange single amino acids in a specific locus. All you have to do is to transform one specific DNA construct into the plant where the nucleus is coded and also the vector is present. And we did this, and our first experiments with Cas9 were unfortunately not so very successful. So it worked, but with hypergenius Cas9 we had an efficiency of 0.1%. So if you have a very motivated PhD student, you might be able to ask him to make the experiment. But for most of the PhD students and for the sake of the lab climate, I would not suggest that we use this technology. And therefore, we decided we have to do something for the poor PhD students that are targeted. Is there any problem? Okay. So, let's continue. Now I'm lost. So in principle, we had this problem that only very motivated PhD students might be able to perform targeting with our procedure, and we have to do something for all the other PhD students. So in principle, we started to optimize the system, and the guy who optimized the system was Felix Wolter. Some of you might know him because he made his master thesis here. I really have to tell you, this is a very excellent student. I'm very happy to have him. It's the best student I have at the moment. And this also reflects the good education at this institute. My congratulations. So I also benefited from it. So in principle, he was the guy who really tried, we tried to improve the system. For this, we used ALS as marker. So here you see the setup. It's the same kind of system. We use a point mutation in the ALS gene. Then we get herbicide resistance, and with the herbicide resistance, we get plants that can grow on the respective herbicide, and this gives us an extremely good readout to optimize the system. And Felix is a very busy guy, and he was very hardworking, and he tested many different things how we can optimize it. And I want to make a shortcut. I only show you the end result of this. we were indeed able to significantly enhance gene targeting in Arabidopsis by exchanging the nuclease. So I told you we had the pyrogenous nuclease in the beginning. Later we found out that the aureus nuclease is much better, so we also used it for gene targeting. This helped, of course. And the other thing which we also changed is in the beginning, we started with the ubiquity promoter I told you, and we ended up with the external-specific promoter. And so if you express the aureus Cas9 in the external-specific promoter, you get a lot of higher gene targeting rates. These are different lines. You see here 6%, 5%, and this is a direct comparison with the lines with our original system. In most lines you don't get anything. Here you get always 1 to 2%, and if you make a good selection, you easily find several lines where you have higher percent rates of gene targeting. So in principle, by using the first cell, the cell where all other genetic information stems from the X cell, and mutates this cell, you are absolutely sure that you can transfer, of course, the change in the next generation, and you have to make it in the most optimal way to use the best molecular scissors you have to induce breaks in all cells possible, and this is achievable by this kind of combination. So we could enhance by 1 order of magnitude the gene targeting efficiency using this technology. Okay, so in principle, mutation works fantastic in all different kinds of plant species. We see light at the end of the tunnel with homologous recombination, with gene targeting, establishing this technology also in many different plant species. But now we can come to the big depressive part of my talk, and this is regulation. Okay, a little bit about the story about regulation. So in 2010, the European Commission asked a committee to evaluate this new double-strand break-induced technologies, and they parted these technologies in 3 different classes: SDN1, SDN2, and SDN3. SDN1 is simple mutation by double-strand break induction. SDN2 was homologous recombination, a small change of 1 or 3 nucleotides. And SDN3 is if you integrate a gene. And in principle, the Commission said, okay, SDN1 and SDN2 are neither identical statistically, a change below 20 nucleotides we will be never able to recognize in a plant genome as foreign. So everything that is below 20 nucleotides should be regarded as a natural change, and only if you insert longer sequences, this is something which you can talk about a transgene. This is like a transgene. But SND1 and SND2 cannot be regarded as transgenic. So in principle, this you have to take into account, and you also have to take into account if you talk about CRISPR-Cas and compare it to GMOs, what are we doing and what are we seeing in the end? So what we are doing is we're using a double-strand break. But double-strand break occur all the time during the life cycle of a cell, so it's a natural process. So whatever we see in the genome after inducing a double-strand break is a natural repair process. There is nothing specific. You never can tell what kind of break was induced. Was it induced by an enzyme? Was it induced by other means? The next point is, of course, if you talk about changes in the genome, you have to be aware how many changes you get in the genome during the life cycle of a plant. And I'm not sure if all of you are aware that if you go in the field, I mean, I discussed this with Andreas Grana, so he should really know. If you go into the field and you take 2 barley plants out of the field, you will find 100 mutations difference already between the 2 plants. So I ask you, how can it be that the 100 of first mutation that was done by CRISPR-Cas can be recognized as something special? And of course, for a time there was this concerns that you had off-site effects, that besides this mutation you induce in the genome, you might also induce other changes. But this is also solves the problem because we have the whole genome technology, sequencing technology, and we can really address this question and can be sure all the changes we induce with this technology. So in principles, all these things have to take into account. And if you take all this into account, there can be only one answer to the question, is a CRISPR-Cas mutated plant a GMO or is it not a GMO? It's just no way to discriminate a CRISPR-Cas induced plant from a natural plant. So the answer, the scientific answer, there's no discussion about the scientific answer, I think. These kinds of plants are not nature identical. There is no way to discriminate them from natural plants. Before this famous date, the European Court rules, the basic idea was in the European community that everybody was allowed, without second source, to use all classical mutagenized material as cultivars. They were not regarded as GMOs, but all plants that ever saw a recombinant nucleic acid had to be regarded as GMOs. This was the difference before the ruling, and there were a lot of statements taken from all the European scientific academies, which all said regulation of SDN1, SDN2 cannot be justified scientifically, and also many other countries, and the list is getting longer. I think Japan joins the club now, Argentina, Canada, the United States, Israel, in all these countries, these kinds of changes are not classified as GMO. But you know, of course, what happened. We were all very shocked because there were a lot of signs before that the decision would go the other way round. But in the end, this is the ruling of the court in July this year. All plants produced by mutagenesis have to be regarded as GMOs and regulated. So this also means chemical and physically mutagenous plants are in principle GMOs. I mean, that's a big difference to before, and they might be allowed to be extended depending on the respective country, if the country wants it, due to their long safety record. So in principle, this ruling is much worse than anything we had before. And the question is, of course, what now? Well, first of all, many people got a heart attack. I also got a heart attack. Yeah? So in principle, there was a big outcry in the whole scientific community, and nobody could understand this because it's against any scientific logic. But of course, if you think about the basis of this ruling, the basis of this ruling is that in principle, the judges took a law, this is outdated, this is 20 years outdated, and the answer, can we change this anyway, is only to try to convince politicians. But before I come to this, I want to just count down some of the consequences we have at the moment. I mean, at the moment, it's really the case that GMO, the status of GMO, there is no scientific definition, but depending where you live, you might have a GMO. If you're in the United States, the same plant is not a GMO. I think that's a very weird situation. And there is no, as I told you, there is no scientific way beyond doubt to discriminate a plant made by CRISPR-Cas as GMO. So if a plant is exported from a country where it's not a GMO, I'm very much interested how this ever can be found out. Obviously, and maybe it's not such important to people working in the breeding sector, but in molecular biology, it's dramatically reducing the chances of our young scientists because we will not be able to perform CRISPR-Cas experiments in companies in Europe. And due to this high costs of regulation, you also kill all the startups, all the small companies in Europe that might have used this fantastic technology at this moment. And we are really pushing this new technology that is only applied by the biggest companies. Sorry to say this. And of course, in the end, all these people that are very happy about this negative outcome of the ruling want what we can supply them with the new technology. I mean, we can supply with the new technology a reduction of pesticides, and we can supply to longer plants, I'm sure, that are adopted to global warming, and all this is blocked by this ruling. So in principle, it's really a very difficult situation, and the only thing I think we can do, and you see here some of these things that are just initiated, we have to, I'm running out of time, but I thought this is extremely important. This is the most important development also for breeding for the next 20 years. So I think we all have to really try to convince the politicians, and as scientists cry out that new laws are made that are up to date, that we can again work on a scientific basis and use the new technology also shortly talk about the future. Forgive me. I mean, it's lunch anyway. 3 more minutes. 3 minutes left lunch. 3 minutes for the future. So the future, I have a view, and I want to share this view with you. My view is that using the new technologies, we will really be able to combine the best alleles of the gene pool of a species in the best configuration. Okay, combining the best alleles of the gene pool of a species in the best configuration. This was not possible till now by classical breeding, out of many reasons. I mean, I give you one example. I mean, we have of course all the seed banks, so we have the material, we have all this genome technology, but to really bring these changes about, there was a very interesting example recently where people knocked out several genes at the same time and coming from a wild tomato to a cultivated tomato by just mutating 6 or 7 traits. And this is work done independently by 3 groups, my good friends Dan Heuters, Shaisha Gao, and Zach Littmann. They all achieved similar things. So you can go in 1 or 2 generations from a wild plant to a cultivated plant by at the same time knocking out a row of specific traits for domestication. So this is one way that clearly demonstrates where we go. Another way, and I want to cut this part dramatically down because I'm running out of time. This is what we are doing. We are also restructuring genomes. So deletions can be induced quite easily. I showed you we also can induce inversions, and I miss now this part because anyway, these results are still unpublished. So, but they're very exciting. So I tell you now, we really are able also to induce inversions in the 10% range in plants, which is a big step forward. But in principle, as a breeder, you of course know that you have this euchromatic region and the heterochromatic region in the genomes, and also you have traits here. You cannot address them by recombination, by crossing, and I think here CRISPR-Cas is the future because we now can induce somatic crossovers, or we might also be able to really directly, and we're working on this, really control inheritance. So in principle, I think if you think about meiotic recombination, there are always breaks induced more or less randomly in the genome. We are at the moment trying to create new hotspots by inducing breaks at specific positions where we want to have hotspots in the genome also during meiosis. And in the long run, of course, you can take out all of this natural breaks. You can do this already now. And CRISPR-Cas gives you the opportunity to induce many breaks at different sites at the same time. So in the long run, I think, and maybe this is not 200 years, this is much earlier, we might really be able to completely control inheritance and plan beforehand every crossing and which alleles we want to combine. I think this is the potential of this technology, and we are far away from this now, but this will work on in the next 20, 30 years. Okay, thank you. And with this, I want to thank you. Wait. I want to thank you, and I want to thank my group, and I also want to thank the European Research Council for funding our work the last 4 years and the last 6 years and the next 4 years. So.
Yeah, thank you very much for this very informative and lively presentation of the topic. Since it's so important, I would like to allow 1 or 2 short questions if there
Holger, in the plant breeding community, genome editing is sometimes discussed a little bit critical for one reason that many traits that breeders deal with are quantitative traits.
I know.
So there are people like Butler who say in public, well, we will do highly multiplexed genome editing to, for example, to remove deleterious mutations. What is your take on this?
I think this is a very good vision. And I also agree with your comments that also this technology will not solve all problems.
Another question? I mean, there will also be the option to approach Holger during the break, obviously, to ask questions. So.
Yeah, but Victor is allowed to ask a question. I know him for 30 years.
Holger, but I'm again critical. I'm fully with you regarding the technologies currently have. And but continues the question what Carl asked about how many knowledge we have for yield gene, for example. Because to do it that, of course, to eliminate some negative effect is a possibility. What we're doing already 160 years by doing the breeding, by selection. But the question how deep we are on yield gene on wheat, for example, how we could use this culprit precise.
For this, we need the knowledge of the breeders, of course. I mean, I cannot supply you with knowledge. I only can supply you with the technology. And I think you are absolutely right. Of course, standing here, I'm extremely overwhelmed by the might of the technology in comparison to everything else we had before. But I think a point that was also clearly made in the last talk is that in the end, only a combination of technologies will bring us forward. So don't get me wrong, I'm not telling you this is the only solution that will be the answer to everything. Absolutely.
Fully agree with you again. Again, I like to get the point is technology is there, but also encourage to give the possibility for young students to go again deeply in quantitative genetics. Because this could be a solution also to find what is behind it.
I think I don't know whether I can answer this question the way you thought about it, but what really astonished me was also that this technology gives us the opportunity to bring variation to specific parts of the genome, and via this way to produce just new kinds of alleles you never had before in specific regions of the genome. And I think this might be, to a certain extent, also helping to answer the points you address. So, in principle, this is a nice thing. Whenever you have a new technology, people have new ideas what you can achieve with this that nobody thought before. And to bring variation, to induce variation in specific parts now, instead of the whole genome, I think is an underrepresented, under-researched area for the moment. I see big advantage there.